In this last video, we'll do cell structure, junctions, and protein synthesis. So with cell components, the plasma membrane is selectively permeable, which means that it allows some molecules to diffuse through the cell membrane and other molecules cannot. And it's a barrier between the fluid inside the cell, the intracellular fluid or the cytoplasm, and the fluid outside the cell, the extracellular fluid. And then inside the cell, we've got the nucleus, which contains the DNA, and the cytoplasm, which consists of the cytosol, which is the gel-like material, and the organelles. So here's the structure of a cell. So make sure you know the cell parts, be able to identify them on a picture, and also be able to tell me the function of the different parts of the cell. And make sure you know table 2.2 from your book. This has all of the different cell parts and the function. You do not need to know about the vaults. I will not ask you about those. You can read about them, but I won't ask you about those on the tests. But everything else should just be a review. I know you've had all of those before. So just be able to recognize if I have a picture, you should be able to identify the cell part and then tell me what it does. The plasma membrane is made out of a phospholipid bilayer that's selectively permeable, which means that it lets some things diffuse through the membrane and other things cannot. We describe the plasma membrane as being a fluid mosaic model, which you can see on this video that's down there. Fluid is referring to the fact that the phospholipids move. So the membrane is not a static structure. And then mosaic is referring to the fact that the membrane is made up of a lot of different pieces or parts. So there's different proteins embedded in the membrane, there's phospholipids, there's carbohydrates, cholesterol. So it's like a mosaic picture, which remember those are made out of different colored tiles. So it just reminded them of that, and so that's what mosaic is referring to. So there's different parts making up the membrane. Okay, and then just looking closer at the plasma membrane parts, we've got the phospholipids that form the bilayer. We've got the cholesterol embedded in the phospholipid bilayer. And remember, we talked about the purpose of the cholesterol. Remember, cholesterol is a steroid, one of the lipids. And remember, the cholesterol is there to help maintain the correct fluidity of the membrane, because you don't want the membrane to be too runny and you don't want it to be too solid. And then we've got proteins in the membrane. The ones that are embedded in the membrane, they're called integral membrane proteins. And if they go all the way through the membrane and they're poking out on both sides, then the integral membrane proteins, we can also call them transmembrane proteins, because trans means across, meaning they go all the way across the membrane. And they can serve different purposes, so they could be receptors, enzymes. A lot of times these transmembrane proteins that go all the way across are going to be used to transport things across the membrane. The peripheral membrane proteins are loosely attached to the inner surface of the membrane, and it's a place for the cytoskeleton to attach. And then we also have some carbohydrates on the outer surface of the cell membrane, and they're going to be attached to the proteins and the phospholipids. So here's just some pictures of the plasma membrane. We've got the proteins embedded in the membrane. So here's an integral membrane protein. Here's another integral membrane protein, and notice that it goes all the way through the membrane. It's sticking out on both sides. So this integral membrane protein would also be called a transmembrane protein as well. And then this membrane protein that's just on the inner surface of the cell membrane, that's a peripheral membrane protein. And that's where the cytoskeleton will attach. And then we've got carbohydrates sticking out on the outer surface of the cell membrane. If they're attached to the phospholipids, we call them glycolipids. And if they're attached to the proteins, we call them glycoproteins. And then we also have some cholesterol embedded in the phospholipid bilayer there. And the cholesterol, remember, is there to help maintain the correct fluidity of the membrane. And then this is just showing you, again, the membrane proteins, just showing you the integral membrane proteins and the peripheral membrane proteins. And then this just is showing you the glycoproteins and glycolipids. And these carbohydrates that are on the surface, the outer surface of the membrane, they will form the glycocalyx. That suffix calyx means covering. And glyco is referring to sugar. So the word glycocalyx just means that our cells have this outer sugar coating on them. And the function of the glycocalyx, one is that it helps hold the cells together. So it acts like a glue. Because if you get sugar wet, it gets sticky, doesn't it? Okay, and our body has a lot of water in it, and so it helps hold the cells together because it's kind of sticky. And then the other thing that the glycocalyx will do is it acts as an ID tag on the cells. So it helps the cells recognize each other and know what kind of cell it is. So for example, on our red blood cells, the glycoproteins on the surface, they will give you your blood type. Okay, so that would be an example of an ID tag. Okay. And then here's just an electron microscope view, just showing you the carbohydrates out here on the outer surface of the membrane of two cells. We've got one cell over here and another cell over here. And you can see how the glycocalyx of each cell comes together and acts like a glue to help hold the two cells together. All right, cell junctions. There are 3 different types. Make sure you know these because these are going to come back later in the course and they will reappear. We have 3 different types. We have tight junctions, and they are going to be found between epithelial cells that line lumens or between skin cells, and they're going to create an impermeable barrier between those epithelial cells. And that prevents things from moving in between those epithelial cells. So if something wants to get beyond that epithelial barrier, it has to go through the epithelial cell to get to the other side. It can't go in between those cells. And then we have desmosomes. These kind of remind me of Velcro. They're like a little spot weld. They're found between skin cells, also between cardiac muscle cells, and also between smooth muscle cells like in the uterus. And then gap junctions. They're little protein channels between cells, and they allow cells to pass messages back and forth between each other for communication, or they can pass nutrients back and forth like in the bone. So we'll find gap junctions in cardiac muscle cells, smooth muscle cells, bone, and then a few neurons will have gap junctions as well. So here's a picture of some tight junctions. So these are some epithelial cells. So maybe this is the wall of the intestine here. And these little brown dots here are the proteins that make up the tight junctions between these epithelial cells. And here's a close-up view. And so you can see that these tight junctions make it so that things cannot go in between these epithelial cells, which is good, because if you have some food or bacteria sitting in here in your intestine, you would not want that to slip in between those epithelial cells and make it into your abdominal cavity, because that would kill you. Okay, this is a close-up view of desmosomes. So you would find this, for example, between your heart muscle cells. And desmosomes, they kind of remind me, like if you look at Velcro, if you look at that up close, the little interlocking fingers of the Velcro, that's what it reminds me of. So these are little protein fingers that hook the cardiac muscle cells together, and that actually keeps the cardiac muscle cells from ripping apart as the heart is beating. So it's good to have desmosomes in your heart. And then these are gap junctions. So gap junctions form little protein channels between two cells, and that allows the cells to pass messages in between them or nutrients. So those are gap junctions. And then lastly, we have protein synthesis. So just remember, it's a two-step process to go from the gene and the DNA to make the protein that the gene is coding for. I also want you to know where this is occurring in the cell. So the first step, going from the DNA to the protein, we're going to take the DNA and we're going to transcribe or copy it. So transcribe or transcription means to copy. Okay, and we're going to do this in the nucleus. And the reason we're doing transcription in the nucleus is because that's where the DNA is. Okay, so that's easy to remember if you just remember that, oh yeah, the DNA is in the nucleus, and that's where we're going to do transcription. Okay, so when we transcribe the nucleus, or sorry, when we transcribe the DNA, we're going to make the messenger RNA, and then the messenger RNA gets sent out into the cytoplasm, and that's where the ribosomes are going to jump on the messenger RNA, and they're going to translate or decode, read the code that's on the messenger RNA and figure out what amino acid to put in what order to make the protein. Okay, so that would be the second step, translation, and that's going to occur in the cytoplasm. Okay, and that occurs in the cytoplasm because that's where the ribosomes are. Okay, you don't find ribosomes in the nucleus. Okay, ribosomes are just in the cytoplasm, so translation has to occur in the cytoplasm.